Greetings! Let us first examine this image of a bird native to the Philippines. Are you familiar with it? Quite beautiful, are they not? Majestic even. Let me introduce to you the Philippine dwarf kingfisher, the Ceyx melanurus. As we go deeper in this presentation, the advent of science and technology gives birth to the foundation of the hypothesized tracking dispersal of Ceyx melanurus. The Philippine dwarf kingfisher lives near streams in lowland woods, with a bright red bill and legs, a white throat, belly, and marked behind the cheek, and black lines along the side of the back. This bird is mostly orange. The head and chest have a pinkish-purple hue to them. Lausan birds have a bluish ear and a darker wing. Like other animals, it helps keep the balance of the ecosystem as it feeds on a variety of smaller animals such as worms, lizards, and invertebrates. What we know based on its taxonomy are the following. Its conservation status is vulnerable as listed by the IUCN. They are found in the Indo-Malayan realm. It is a species endemic to the Philippines with a decreasing population trend due to extensive lowland deforestation throughout its range. This study is important to contribute new information about the species for the advancement of further research and for conservation plans. Each and every one of us is called to our responsibility to learn more and invest in these species. The Philippine scenario chosen is the Ceyx lepidus phylogeographic publication by Anderson et al. For its rationale, the availability of data for the Ceyx genus is still limited and other related studies are still ongoing. With this, the study was able to expand the information with regards to the species Ceyx melanurus and its subspecies through phylogeographic means. Each taxon in the Ceyx lepidus complex is monophyletic and substantially diverged from all the other taxa. It is important to note that from the results of the study, the highest diversity of dwarf kingfishers occurs in the Philippine archipelago. Now, for its short introduction, Southeast Asia's islands and Western Pacific are home to some of the world's most phenotypically varied avian species complexes, and Ceyx lepidus, or variable dwarf kingfisher, is one of them. The related species were studied through mitochondrial and nuclear DNA sequences to track their relationships. Lastly, the objectives of the studies are, first, to reconstruct the phylogeographic relationships of the variable dwarf kingfisher using DNA sequence data, and secondly, to discuss species limits and make taxonomic recommendations to treat all 15 subspecies of Ceyx lepidus.
Here is an overview of our concept map.
The main objective of this study is to learn more about the evolutionary process that led to the geographic spread of the dwarf kingfisher Ceyx melanurus in eastern Visayas and Mindanao. The goal of this study is to establish the phylogeographic relationships of the C. melanurus using genomic data and geographic distribution data. As cited in the paper of Anderson et al., the available information with respect to its genetic structure and morphological variation for the C. melanurus and its subspecies are still limited and ongoing. Thus, the following are the specific objective of this research: to expand the current available data, obtain samples through fieldwork, and publish the results in scientific databases such as the GenBank database.
For our proposed research, our alternative hypothesis states that the genetic diversity among the subspecies of Ceyx melanurus is correlated to its dispersal. Meanwhile, for the null hypothesis, there is no relation between the Ceyx melanurus genetic diversity and its dispersal. We would test this hypothesis by mapping the species recorded sightings in eastern Visayas and Mindanao, as well as obtaining their genetic material and using it to detect molecular variations and potential gene flow between subspecies in different regions.
So for the geographical data set of the methodology, we first start with locating the distribution of the Ceyx melanurus subspecies. Now once we are done with locating the said species, we proceed with the acquisition of occurrence and coordinates for the kingfisher. This will be done through actual field studies and researching databases such as GBIF and MoveBank. Once we have acquired the necessary geographical data, we proceed with the utilization of QGIS and DivaGIS software. This will be used as a platform to plot the gathered coordinates. Then we proceed lastly by recording, analysis, and the interpretation of data.
For the sampling methods, first we will establish the areas where we will obtain our samples from the kingfisher subspecies, after which we will survey and identify the individuals from which we will collect the sample by swabbing the inside of their cheeks. After sample collection, DNA sequencing will follow. This includes DNA extraction and purification and PCR amplification, which is simply the duplication of DNA. This will allow us to detect and identify the mitochondrial DNA and microsatellite DNA sequences needed for genetic analysis. Last, we will utilize the Arlequin and DivMigrate software to examine DNA in order to determine molecular variants and gene flow respectively. Once all these data are collected, we will analyze and interpret the subspecies genetic makeup and its connection with dispersal.
This research focuses on investigating the evolutionary process that was responsible for the geographic distribution of dwarf kingfisher in the Philippines. Research, fieldwork, and species observations are limited only within Eastern Visayas and Mindanao. In addition, there are multiple endemic species of dwarf kingfisher in the Philippines. However, this study focuses on C. melanurus and its subspecies specifically. Moreover, the available track records of C. melanurus occurrence from the GBIF database are only limited from the year 1869 up to 2021. The following are the expected outputs when the research study is completed. Available information for C. melanurus is still lacking. Fieldwork to obtain data such as geographic data and genetic data will be done in the study. This data will be utilized in identifying and explaining the specific evolutionary hypothesis that was responsible for the past to present geographic distribution of C. melanurus. And this research will be publicized in journals, making the findings more accessible to researchers. In addition, the data obtained from the fieldwork will be published in databases such as GenBank, for it to be also accessible to researchers. This new data, knowledge, and information gives the opportunity for future researchers and scientists to further deepen their knowledge about the C. melanurus. Moreover, this study can be utilized as a reference for future researches and for future conservation plans.
And now for the workforce and specialist of our team, we are comprised of our field head biologist, Ms. Locre, Mr. Africa as our laboratory head biologist, and of course our very own geneticist, Ms. Verba. We also have our geographer and cartographer, Ms. Maranan, and of course yours truly as your avian biologist. We also need the following expertise such as field biologist, wildlife photographer, technicians, local area experts, and of course our geographical surveys.
The budgeting of this experiment ranges from 4.6 million pesos to 5 million pesos. The largest expenditure for the experiment would be the personal services, or specifically the salary of the specialist, with the expectation of a 10-member research team including the specialist, and computed with the average yearly rate of a Philippine researcher. The second and third expenses to be discussed are the travel and rent expenses, which the travel expense include both air, sea, and land transport, which was calculated to the destinations using PAL services or Philippine Airline, and the various rental services. The renting of lodges was calculated with TripAdvisor on the sites of Samar and Cagayan de Oro, Mindanao. Outlay refers to the non-expendable equipment for fieldwork and laboratory work, while supplies and materials are the expendable ones. Allowances for daily sustenance only refers to the 6 months of fieldwork, and communication refers to the phone and internet services while in the fieldwork, while transportation refers to the packaging of molecular specimens and our equipment, while indirect costs refer to the payment of permits for the fieldwork. The total minimum budget is around 4,556,070 pesos.